Good morning, students. Good morning, students. Am I audible?
Yes, ma'am. Good morning, ma'am.
Yes, ma'am. Good morning, ma'am.
So, okay, recording has started. So, today we are going to discuss about somatic hybridization, basically, right? So, last time we discussed a technique which was called as a somatic embryogenesis, also that we discussed, right? So, somatic embryogenesis is different than somatic hybridization, as we'll study today. Like, so in somatic embryogenesis, a single cell or a group of a cell, they follow a certain pathway which leads to a generation of some non-zygotic embryos, right, which are again able to produce a whole plant. So this we have studied already, and today we are going to discuss about somatic hybridization. So in somatic hybridization basically is about the we isolate the protoplasts of the cells, plants, protoplasts from the plants, right? So what we do in somatic hybridization, we in this we can take protoplasts from two different plants and we can fuse them. So in this way, a hybrid will be generated, right? A plant will be generated which will have a characteristics of both the plants or plants from which we have taken a protoplast, right? So we understand that protoplasts are the plant cells without the cell wall. So there is a process basically that we isolate protoplasts from the plants, then we do the fusion of the protoplast of the different plants, like from maybe plant A and a plant B, which whatever characteristics we require, and then there are a lot of different ways by which a fusion of a protoplast can be done, and then a hybrid plant is being generated, and then we need to select the hybrid plant also, right? Because number of plants are being generated by this combination. So we need to select those hybrid plants which has the desired characteristics. So that process we are going to study today in detail, right? So when we talk about somatic hybridization, it basically this technique involves three steps or three different processes. First is that how do we fuse the protoplast? We need to fuse the protoplast. So first is the fusion of the protoplast. Second is the whatever hybrids we get out of those fusing protoplast, we need to select those hybrids. So we are not going to take all the hybrids, and the selection of the hybrid also involves a lot of processes, that how we know that this particular hybrid plant has both the taken a gene from the protoplast A also and protoplast B also. So how do we identify that? There are certain techniques available for that also, right? Then selection of the hybrid plant, then identification. In the end, when those plants are being grown, and then we need to identify those plants, that which plant we need to move with the further, whatever the characteristic we require is present in which plant, and how do we move further to taking up those plant in the fields, right? So we all know, right, the conventional methods of plant breeding is sexual hybridization. We know it occurs in the nature, and the variations are being produced in the plants. But the major limitation of sexual hybridization is that it can be performed within a plant species, or they should be closely related, right? They should be related species. So this somewhere restricts the improvement that can be done in the plant if we want to take some plant from some other species which are not related at all, right? So sexual incompatibility happens when we do that, or that process is taking place in the natural environment or in the nature, right? So species barrier for plant improvement encountered in sexual hybridization can be overcome by somatic cell fusion. So that can form a viable hybrid. So somatic cell fusion technique that can be, we can use this can lead to the hybrid plants which we can be generated. So somatic hybridization broadly involves in vitro fusion of isolated protoplasts to form a hybrid. So we isolate a protoplast from the plants, we fuse those protoplasts to form a hybrid cell, and then subsequent development to form a hybrid plant. So this is about the somatic hybridization. So plant protoplast, we know they are being They are of immense utility in somatic plant cell genetic manipulations and improvement of the crops. So thus, these protoplasts, they provide a novel opportunity to create cells with new genetic constitution. So, for example, in a plant A, the plant A, which has a characteristic, maybe it is resistant to a particular disease. Then plant B have a characteristic, maybe it is a high yielding variety. Now we want that both these characteristics should be present in one plant. So for that, this process is very useful, and by this process we can generate such plants, right? So this process basically involves fusion of protoplasts, selection of hybrid cells, and then identification of the hybrid plants. First is the fusion of the protoplast. So there are a lot of methods, a lot of chemical methods, and so a lot of mechanical methods which are available for fusion of the protoplast. So we know the protoplasts are the cells basically which do not have any cell wall. So in vitro fusions become easy by this method. So there are no barriers of incompatibility for the protoplast fusion. The protoplast fusion that involves mixing of protoplast of two different genomes can be achieved by spontaneous, mechanical, or induced fusion method. So there are spontaneous method, there are mechanical method, or there are certain chemicals which we can use to induce the fusion processes. So first is a spontaneous fusion. So as we all know that cell fusion is a natural process. For example, it is being seen in egg fertilization, and during the course of enzymatic degradation of cell walls, some of the adjoining protoplast can also fuse to form homokaryocytes or homokaryons, right? So these fused cells may sometimes contain high number of a nuclei, right? They contain more than one nuclei also. So this is mainly because of the expansion and subsequent covalence of plasmodermal connections between the cells. So the frequency of homokaryon formation was found to be very high in protoplast, which was isolated from dividing cultured cells. Now, spontaneously fused protoplasts, however, cannot regenerate into a whole plant except undergoing a few cell divisions. So we need to do something, right? A mechanical fusion or some other ways of a fusions. So the mechanical fusion, the protoplast can be pushed together mechanically to fuse. For example, protoplast of lilium and trillium in enzyme solutions can be fused by gentle trapping in a slide. So mechanical fusion, but there's a drawback here that fusion may damage the protoplast by causing injuries to the protoplast. So this is again not a very appropriate method. Then we have an induced fusion. Now, freshly isolated protoplast can be fused by induction. So there are several fusion-inducing agents which are collectively referred to as a fusogens. So fusogens are the chemicals or those agents which can be used for the protoplast fusion or which can lead to the induction of the fusions, right? So these are sodium nitrite, polyethylene glycol, polyvinyl alcohol, lysozyme, then concovalin A, dextran sulfate, fatty acid esters, or some electrofusions are also there. So some of the fusions, we will discuss some of the fusions here, that how they are being used. So this is actually a process of a protoplast fusion. So we'll take maybe two different strains or two different plant. We will take a protoplast, right? Then we'll treat them with some fusogens or those chemicals that we discussed, that we named in the last line, and then the fusion will take place and there will be a fused protoplast which will be obtained. So treatment with the sodium nitrite. So sodium nitrite is one of the commonly used fusogen, and here the isolated protoplast, they are exposed to a mixture of 5.5% sodium nitrite in 10% sucrose solution. Now then we incubate. Incubate means we keep them for 5 minutes at 35 degrees Celsius, followed by the centrifugation after that. Now the protoplast pellet that we obtain is kept in water bath at 30 degrees Celsius for about 30 minutes. During that time, the fusion of the protoplast will take place. The sodium nitrite treatment results in a low frequency of a heterokaryon formation, particularly when the mesophyll protoplast are used. So this is one drawback here that we are facing. Then we have a high pH and high calcium ion treatment. This method was again used for the fusion of tobacco protoplast, and it is being used for in other plants also. Here we incubate the protoplast in a solution of 0.4 molar mannitol, which contains 0.05 molar calcium fluoride at pH 10.5, and temperature should be 37 degrees Celsius for around 30 to 40 minutes. Here the protoplast, they form aggregates, and the fusion usually occurs within the 10 minutes only. And this method, around 20 to 50% of the protoplasts are involved in the fusion. So the rate is higher in this case. Then we have a PEG, polyethylene glycol treatment. Again, this method is most commonly used because of its success rate. The success rate is high in this case. So here also the isolated protoplast in culture medium, they are mixed with equal volume of 28 to 56% PEG in a tube. Now this PEG, it enhances the fusion of protoplast in several species. We shake this tube and then we allow it to settle. The settled protoplasts are washed several times with the culture medium before we use them further. So this PEG treatment method is widely used for protoplast fusion because it has a number of advantages. It results in reproducible high frequency of heterokaryon formation. It is less toxic to the cells. There is a reduced formation of binucleate heterokaryon. The PEG-induced fusion is a non-specific and therefore can be used for a wide range of the plants. So it is not only for a few plants. It can be used for a number of the plants because it is a non-specific method. Right? It is not only used for a few plants only, right? Then we have Electro Fusions also. So in this method, as the name suggests, we use electric field for the protoplast fusion. So when the protoplast, they are placed in a culture vessel which is fitted with the microelectrodes, and then we apply the electric shock, the protoplasts are induced and they fuse. So this is again a very simple, quick and efficient method, and has been commonly used. Further, the cells formed due to electrofusions, they do not show cytotoxic responses, as is the case of certain other fusions, which is fusogens such as PEG also. So they can show some cytotoxic responses. The major limitation of this method is the requirement of a specialized and costly equipment. So electrofusion is definitely being done by some electroporator or some other equipment which come for generating those kind of a shock at a particular voltage, right? So we need to have that particular equipment for this method. Then there's a mechanism of fusion. So how does the fusion takes place? So this again involves three phases such as agglutination, plasma membrane fusion, and then the formation of heterokaryons. So first is a agglutination or adhesion. So when two protoplasts, they are very close to each other, or they are in close contact with each other, adhesion will take place. The agglutination can be induced by fusions such as PEG, high pH, or a high calcium ion concentration. Then second is the selection of a hybrid cells. So about 20 to 25% of the protoplasts are actually involved in the fusion. So after the fusion process is complete, this protoplast population will consist of a heterogeneous mixture of unfused chloroplast of heterokaryons or homokaryons also, right? So we need to select the hybrid cells. We want heterokaryons. We don't want unfused chloroplast. We don't want homokaryons because we are interested in the heterokaryons or the hybrid cells. The commonly used method employed for the selection of hybrid cells are biochemical, visual, and cytometric method. So these are the commonly used methods which are being employed. Biochemical method for selection of hybrid cells are based on the use of some biochemical compound in the medium. So these compounds, they help to sort out the hybrid and the parental cells. तो ये उस तरह के compounds होंगे जिसमें parental cell नहीं grow कर पाएंगे, and only minimal medium बोलते हैं. We always call them as a minimal medium. So only the hybrid will grow on those medium, not the parental cell. So in this way, we will be able to distinguish, right, based on their differences in the expression of the characters. Then drug sensitivity and oxalotrophic mutant selection methods are here. For example, drug sensitivity. So this method is sometimes used for selection of hybrid of two plant species if one is sensitive to the drug and other one is not sensitive, right? So, for example, the protoplast of Petunia hybridae, which species A can form macroscopic callus on the MS medium, but are sensitive it or means inhibited by actinomycin D. Whereas the Petunia parodi protoplast species B form small colonies but are resistant to actinomycin D. So both have a different type of the requirement. So when these two species are fused, the fused protoplast derive both the characters, right? The formation of macroscopic colonies also and resistant to actinomycin D on the MS medium. So this helps in the selection of the hybrids. Now the parental protoplast of both the species, they will not grow, right? Because उनमें दोनों characteristics नहीं है, so they will not grow. The protoplast of Petunia parodi will form very small colonies, while that of Petunia hybrida are inhibited by the actinomycin D. So only those colonies will be grow, or only those will grow which will have both of these characteristics, or which are the hybrid of both of the parents. So in this way, we'll be able to select. For example, here the species A Petunia hybrida, the species B is Petunia parodia. So we are isolating protoplast from both the species. We are fusing them. Now the culture medium will be supplemented with actinomycin D, right? So what happened? The Petunia species A होगी तो वो बहुत small colonies form करेगी, no further growth. Whereas in case of Petunia parodi, there will be no growth and no divisions because it was sensitive to this, right? Only the fused product which has the hybrid of both of these, they will form bigger colonies and they will form further growth and the callus formation will take place. So this is how we are going to select them, right? Hybrids. Then we have oxalotrophic mutants. Oxalotrophs are the mutants that cannot grow on a minimal medium, and they therefore require specific compounds to be added to the medium. For example, nitrate reductase deficient mutants of Mycosina tabacum are known. So the parental protoplast of such species cannot grow with nitrate as the sole source of the nitrogen, whereas hybrids, they can grow. So this is how we are going to separate them, right? So the two species of nitrate reductase deficiency, one which is due to the lack of apoenzyme and other which is due to the lack of molybdenum cofactor are known. So the parental protoplast cannot grow on nitrate medium while the hybrid protoplasts can grow. So this is how we are going to separate them. And for example, here also, this is the example of tobacco, the species A and species B. We isolate the protoplast, again fusion will be done. The minimal culture medium with nitrate will use. So here in species A, we will not see any growth or any division. Again, in species B alone, there will be no growth or no divisions. But in the hybrid one, the fused one, which will have both the characteristics of species A and B, will be able to show colonies and the callus formation. So this is how we are going to again separate them, right? Then there are certain visual methods also, right? So visual selection of hybrids, it is though very tedious, but it is very efficient. So in some of the somatic hybridization experiment, the chloroplast deficient protoplast of one parent are fused with the green protoplast of the other parent. So the visual selection methods are not feasible for all the types of the protoplast fusion we are doing. But here they've given an example that in which one is the chloroplast deficient protoplast of one parent is fused with the green protoplast of the other parent. So in this, will be visually be able to differentiate. For example, the heterokaryons are bigger, they are green in color, while the parental protoplasts are either small or colorless. So this is only in such cases visually we'll be able to differentiate. So further identification of these heterokaryons has to be carried out to develop specific hybrid plant. Then visual selection coupled with differential media growth. So there exist certain natural differences in the sensitivity of the protoplast to the nutrients of the given media also. So thus some media can selectively support the development of the hybrids, but not the parental protoplast. So we can do that way also, right? For example, here the species, this is the example if visually we can differentiate. For example, species A is a green, whereas species B is an albino or a non-green. So we'll isolate the protoplast from them, we'll fuse them, we'll culture them in a selection media. So in case of a species A, small or a green color colonies will be formed, but further growth नहीं होगी because it is a special selection media जिसमें parents single नहीं grow करेंगे, right? And in other case white cell colonies form होंगी because it is non green or albino, and white callus होगा. So we don't need white callus also because we are interested in both the characteristics. So the center one जिसमें green से color की colonies होंगी और green callus होगा, will go for that, right? So this can be done again by the visual selection. Then there is the mechanical isolation. So visually identified heterokaryones under the microscope can be isolated by the mechanical means. So this again involves the use, there's a special pipette which is called Brumont pipette. So we use this pipette to suck them, those kind of a photoplast. So then the so isolated heterokaryones can be cloned to finally produce the somatic hybrid cells. The major limitation of this method is that each type of hybrid cell requires special culture media for its growth. So we need to check this beforehand, that what kind of a culture media they require, right? Then there is identification of hybrid plants. So development of hybrid cells followed by generation of hybrid plant require clear proof of genetic contribution from both the parental protoplast. This will also see that how we'll do that, how we'll know that this plant has the genetic contribution from both the parental protoplast, not only from the single one. So first is the morphology of the hybrid plants that we are going to see. So morphological feature of hybrid plants, which are usually intermediate between two parents, can be identified. For example, the vegetative and floral characters we'll consider, right? So such as what kind of a roots are there, what kind of a leaf are there, what is the shape of the leaf, what kind of a flower is there, what is the shape of the flowers, color, seed capsule. All these factors we'll take into consideration. Then the somatic hybrids such as tomatoes and topetos, right? Tomatoes and topetos, basically tomato and potatoes. These were the plants which were fused of potato and tomato, right? So they were certain plants a few years back were being produced that they will produce potatoes underground tubers and tomato on the top, right? So these plants were also generated. So these can be identified. We'll know. देख के पता चल जाएगा, right? Although the genetic basis of morphological characters has not been clearly known, so intermediate morphological features suggest that traits are under control of multiple genes, right? We'll know that. It is preferable to support hybrid morphological characters with evidence of genetic data. So morphologically, we have seen that, okay, there are certain variations, there are certain combined characters, but then we'll need to verify this genetically also. We need to see the genetic makeup of the plant also in order to know that it is a hybrid. So there is an enzyme analysis of the hybrid plants. So the multiple forms of enzyme catalyzing the same reactions, these are called as isoenzymes, right? So electrophoretic patterns of isoenzymes have been widely used to verify the hybridity, to verify whether there is a hybridity or not. So somatic hybrids, they possess specific isoenzymes or certain enzymes of one or other parents or both the parents simultaneously. So they are having those kind of isozymes. So there are many enzymes possessing unique isozymes that can be used for identification of somatic hybrids. For example, amylases हो गया आपका, esterase हो गया, aspartate हो गया, isoperoxidase हो गया, right? So all different kinds of enzymes. So if the enzyme is dimeric, that is having two subunits, somatic hybrids usually contain isoenzyme with the intermediate mobility properties. So what happens? The isozymes are often variable within the same plant. Change हो सकते हैं. So therefore, it is necessary to use the same enzyme for each plant. Parents के लिए भी और somatic hybrids के लिए भी from specific tissues with the same. So we are using the same kind of an enzyme to see it. So there are the molecular techniques also. So what are the molecular techniques? Molecular biology, definitely the molecular biology has evolved so much that we are totally dependent upon the molecular biology techniques to see any kind of changes in the genome of a plant or viruses or pathogens. So they are the easily available techniques which one can follow and which one can see. For example, differences in the restriction pattern of chloroplast and mitochondrial DNAs can be done. So there will be a difference in the pattern. So molecular markers such as restriction fragment length polymorphism or amplified polymorphism, and then amplified fragment length polymorphism, then there is RAPD, number of methods. So ये सारे methods में क्या करते हैं, they just they just you run your samples in a gel, you restrict with a different kind of enzymes, and then you see the pattern. Then you see the pattern in each isolated. We are talking about the Protoplast here, hybrid plants here. So we'll see the pattern in each hybrid plant that we have generated. So any variations or any similar kind of a pattern will show us whether we have obtained some kind of a genetic variability or not, right? So molecular markers are very commonly used. PCR technology is being used, but PCR we are going to amplify, we are going to clone, and then we are going to sequence. So after sequencing, we'll get the whole genome of the plant or whatever gene we are focusing on. So that there also we can compare the sequence of the plant with the parents of the plant, that how much we have got from one parent, or there are BLAST2 sequences or BLAST10 sequences. There are softwares available. We'll put the sequences and we'll see what kind of a changes we see in them, right? How much percent similarities with the A or how much percent similarities with the B parent. So all those things can be done by the molecular methods or a PCR technology and the sequencing techniques, right? Then there are symmetric and asymmetric hybrids also. So if the chromosome number in hybrid is the sum of the chromosome of the two parental protoplast, the hybrid is said to be symmetric. And the symmetric hybrid between incompatible species are usually sterile. They are not, they are not going to produce seeds or a flower. Asymmetric hybrids have abnormal or wide variation in the chromosome number than the exact total of the two species. And these hybrids are usually formatted with full somatic complement of one parental species, with all or nearly all of the chromosome of the other parental species are lost during the mitotic division. So that's why they are asymmetric. Asymmetric hybrids can also be regarded as chybrids, but for the enterogrust gene. So chybrids क्या होते हैं? Basically, hybrids, cytoplasmic hybrid, where the nucleus is derived from only one parent and cytoplasm is derived from both the parent. Such plants are described as the hybrids. So the phenomena of formation of hybrid is also regarded as hybridization. And these are basically, these kind of plants are being produced when protoplast from two phytogenetically distinct species are fused. That means they are not similar species. So in these cases, we can lead to the formation of hybrids. And genetically, hybrids are hybrids only for the cytoplasmic traits. So they have majority of the cytoplasmic traits, right? So what are the applications of somatic hybridization? Again, we came to the disease resistance, and this one application is common from all the tissue culture applications that we have studied till now. So several interspecific and intergeneric hybrids with disease resistance have been created, and many disease resistance gene, example tobacco mosaic virus, potato virus X, or a club rot disease, could be successfully transferred from one species to the another by this method. For example, the resistance has been introduced in tomato against it should be tomato mosaic, not tobacco mosaic virus, not tomato. It should be tobacco mosaic virus, spotted wilt virus, and the insect pest. So this is a very good method to introduce disease resistance into the varieties or to develop a pathogen resistant plants by this method. Then environmental tolerance, the genes responsible for tolerance of the cold, frost, and salt could also be successfully introduced through somatic hybridization. Example, introduction of cold tolerance gene in the tomato, which are being done. Just a moment. So another factor use is the environmental tolerance. That means the environmental stress tolerance genes can be introduced into the plants by this method. Then quality, this is just table is showing the selected list of genetic traits which are transferred through protoplast fusion in the crop plant species. For example, the first one is a genetic trait in a new plant, resistance to pathogens. For example, Nicotiana tabacum and another species of Nicotiana is a tobacco. The plant was developed resistant against tobacco mosaic virus, then they have developed against potato virus X also. Then there's a frost tolerance also. Then there's a potato leaf roll virus also. There is some varieties against the nematodes also, right? And then the frost and salt tolerance. So this is being used. Then again, they have also generated plants with the traits or a quality or a character such as high nicotine content in a Nicotiana tabacum, and by crossing Nicotiana tabacum and Nicotiana rustica, two varieties, right? Again, the cytoplasmic hybrids or hybrids are being developed with the agronomic character such as resistant to streptomycin, or cytoplasmic male sterilities is being introduced in certain plants, right? So this method has been widely used uh, to confer resistance to the plants, or to improve the quality or character of the plant, or to introduce any kind of agronomic character in the plant, right? Other application of somatic hybridization, again, it has helped to study the cytoplasmic genes and their functions. Again, this is successfully used in a plant breeding program. So this method is being very commonly used. A protoplast fusion will help in the combination of mitochondria and chloroplast to result in a unique nuclear cytoplasmic genetic combinations. And somatic hybridization can done in plants that are still in juvenile phase. So we can and plants in a juvenile phase can be used for somatic hybridization. Then protoplast transformation with traits like nitrogen fixation by incorporating exogenous DNA, followed by somatic hybridization, will yield the innovative plant. So transgenics, we can also lead to the transgenic plant development by using this kind of a technique. Then we know that every application, they have positive points, but they have a certain drawbacks also, right? So limitations are always associated with any kind of a techniques. So here again, it does not always produce plants that give fertile and viable seeds. So we need to again select plants that are able to produce viable seeds and fertile seeds, right? Regenerated plants which are obtained from somatic hybridization are often variable due to somaclonal variation. So somaclonal variation is here also, right? And that is not a positive point that we don't want here, right? Then protoplast fusion between different species is easy, but production of viable somatic hybrids is not possible in all the cases. So we need to look for this also. And some of the somatic hybrids, particularly when produced by fusion of taxonomically different partners, they are unbalanced and they are not viable. Then there are limitations in the selection methods of hybrids also, and many of them are not very efficient. And there is no certainty as regards the expression of any specific characters in somatic hybridization. So after, at the end, we'll need to follow all the guidelines and all the steps to see the hybrids, whether desired characters are being introduced or not. So there's not like 100% surety that it is going to, characters are going to be in the desired plants or the plants or hybrids that we have selected. Right. So these are some of the drawbacks of the somatic hybridization. But every technique have a positive and a negative side also. So it's fine. Right? We always look for the positive side and we try to exploit the best out of the techniques. So we definitely in every kind of a experiment we do, we get see, for example, it's something like that. When we are doing some experiments, there are certain positive results and certain negative results also we get. So we focus more on the positive side rather than on the negative side. So this is the thumb rule for performing any kind of experiment or a research experiment also, that yes, we know that there are going to be some drawbacks or some negative points, but then there are going to be many positive points and many positive things also which we are looking forward for, right? For when we talk about the uh, the sure shot method for introduction of any kind of a characteristic which is desirable is transgenic till date, because in the transgenic we know which gene we are incorporating in a new plant, and we also know the corresponding factor which is associated with that gene which is going to be expressed in the new plant. So transgenic is only the sure shot way or 100% way that by which we know that plants that we are going to generate will have that particular characteristic, so that gene will be expressed. Other methods, whether they are the breeding methods or other methods, tissue culture methods, we are still dependent upon the selection, right? We don't know. We are going to get number of the plants, number of the mixed plants, and out of those plants we need to select. For example, in hybrids also we are going to get number of the hybrids, but then we need to select those hybrids which has the desirable traits and which we need to use further, right? So that is one positive point of a transgenic, that there we don't need. In the beginning, we know that we are going to manipulate this gene, we are going to express this gene in this plant, and this is going to happen. This is the best positive side of a transgenic plant production, or any transgenic generation, whether they are for disease resistant or whether they are from any other characteristics. So this is how we say when we talk about the conventional breeding method and when we compare them to the transgenic. So transgenic ones, they seems to be more promising as compared to the conventional methods of the breeding, right? Okay. So this is all for today's lecture. The somatic hybridization we are done, and tomorrow you have exam of a plant tissue culture, right? The first was for tissue culture, and the third is for your, what is that? Your third is your plant pathology, right? So tomorrow we don't have any tissue culture class, but I'll publish one tissue culture class tomorrow to take the exam, maybe just alongside the pathology class, so that we can have two classes, right? So to prepare for a tissue culture will not include today's lecture for tomorrow's exam. We'll exclude this. Before this, till last week, whatever we have done, just go through those things, and on the whole syllabus till last week is going to come in the exam, right? Fine. Okay. So another thing, that in plant tissue culture we have one home assignment also. So very soon I'm going to publish the home assignment also. Since presentation we are taking in pathology, so not in tissue culture, we are not going to, and we don't have that much time also. We need to complete the course by February. So no presentations in tissue culture, only home assignment and assessment in a class test. So that we are going to do, right? Fine. Okay. So let's take the attendance and then we can conclude the meeting. Okay, fine. So please switch on your cameras for attendance. Adish Mishra.
Present, ma'am.
Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi.
Present, ma'am.
Ishita Arora.
Present, ma'am.
Kshitij Singh. Manaswani.
Present, ma'am.
Namrita.
Present, ma'am.
Nehal.
Present, ma'am.
Rupal.
Present, ma'am.
Sahiba.
Present, ma'am.
Sanjana.
Present, ma'am.
Shakti.
Present, ma'am.
Shivani.
Present, ma'am.
Tanvi.
Present, ma'am.
Pridev.
Present, ma'am.
Vivek. Yukta Yuvraj.
Present, ma'am.
Okay, so I'll see you tomorrow. Devanshi, आप मेरी class fix करवा देना, extra class कल के लिए एक. I'll talk to you after the class, and let's see in which class we'll have exam, right? Okay, students, I'll see you tomorrow. Bye bye.
Thank you, ma'am.
Thank you, ma'am.